What's up?
Use the mic.
Use the mic. Hi, everybody. Hello. That's tall. All right. Hi, everybody. My name is Anna, and I'm here today to talk about our work dancing with the dust devil, examining the lung microbiome of the Sonorodescent mammals and the effect of Coccidioides. So, can I take this out of here? Is that all right? Cool. So we all know that Coccidioides is capable of infecting mammals, and that, in fact, all mammals in this slide right now were reported at some point to have coccidiomycosis. I just look at my boss right now, so might avoid that. Uh, the question here is, what is the role of those mammals in the life cycle of Coccidioides? So all those papers in there will lean one way or another. And in 2019, Dr. Taylor and Dr. Barker came up with the endozoan small mammal hypothesis, reservoir hypothesis. And in the small mammal endozoan hypothesis, the mammals, the hosts, they would be reservoirs. So what's the benefit for Cocci? Cocci would access the nutrients from the carcass as soon as the mammal host is gone. In other words, Coccidioides can behave as a pathogen, but also it could be in the lungs without causing coccidio, because you said you have to say it, I can't, coccidioidomycosis. So in a way, or what really is going on, Cocci is there, but just as part of the microbiome. What's microbiome? Microbiome is a collection of microorganisms that exist in an environment, in this case, the lung. Up until not that long ago, we thought that the lung was sterile tissue, but now we know it's a very dynamic environment. You know, things are changing in the time of breath. And for that reason, it's very challenging to study. There's transient species, and they're like the species that are part of the established microbiome. One thing that we do know is that infections are expected to cause disruption of that microbiome, and likely that will lead to a lower diversity as infection progresses. So, you know, if Cocci was causing disease, then the more Cocci, the less of other microorganisms. So on the flip side of that, if Cocci is an endozoan, not causing disease, then we won't expect changes in microbiome. And notice that now I'm saying mycobiome. It's because on this study, we are only looking at the fungal portion of the microbiome. So I'm going to say mycobiome starting now. So to try to test that, we first need to screen samples for Cocci. Then we describe the lung microbiome. And then, assuming all goes well, we can compare the microbiome diversity between positive and negative samples. So what samples do we have? Oopsies. Uh-oh. What samples do we have? We have a set of samples from Tucson, Arizona. Those are 26 lung samples and several different species. We also got soil samples to go with that. And then we have a set of samples from Mesa. They're all cactus mouse. And we don't have soil samples for that site because our lab has been doing a lot of work on that site. So we already have extensive data on the soil samples. I will have a little spoiler here, but you saw Marika speaking about that last year as well. And there's a bunch of posters about that site. So please go check it out. Okay, so oopsies, man, this is kind of hard. Okay, so first thing, we did a DNA extraction. So for the soils, we used the soil kit. But then for the lungs, this is a study of opportunity. So we did use two different extraction methods. And for that reason, so Tucson, we used a blood and tissue kit. And then for Mesa, we used a phenylchloroform extraction. So for that reason, we decided to keep those two data sets separately. So analyze those two data sets separately. Once we extracted the DNA, we screened it for Cocci using Coccidioides. Then we looked at the microbiome through a Illumina amplicon sequencing targeting the ITS2 region. And then finally compared the diversity through alpha and beta diversity. And We also use this database called Fungal, plus some of our own research, to try to do functional assignments to the species of the microbiome. So if they were previously known to be associated with animals or plants or dung or whatever. So let's see what we found out. So in Tucson, we found out that about 10% of the samples were positives, 2 out of the 4 boroughs that we sampled. And then we got about 12 genera per sample. And in this case, we got 5 samples to be positive for cocci across 4 different species. As you can see in the plot, cocci is not in the top 10 or top 5. Sorry, it is on the top 10, but not in the top 5. And you have other genuses that were known to be associated with mammals, for example, Ahillomyces. In the Mesa side, we got about 16 genera per sample, and the top 10 corresponds to 94% of the sequences we identified. But here, we had a surprise. All samples turned positive. So for that reason, we are not able to do any further comparisons with this data set because you can't do positive against negative when you don't have negative, which is a new problem, I feel like, when you're screening small mammals for cocci. So I feel special. Okay, what we found out with the functional assignment, actually, most of the genuses we found on our samples were previously identified to be associated with mammals. So about 89% of the sequences in Tucson and 88 in Mesa were already for genera that were reported to be pathogens or commensals or to have any sort of associations with mammals. Okay, diversity comparisons. Remember, this is just for Tucson, no Mesa site here. We didn't find any effect of Coccidioides on the microbiome diversity, neither for alpha diversity or beta diversity. We did look at the effect of host genus or family, and that do explain some of the variation in beta diversity, but not in alpha diversity. We get similar results when we use the Fungal parse data, so species, genuses that were known to be associated with mammals before. So probably transient species were not affecting our analysis for this data set. So what did we learn in this study? First, Coccidioides prevalence in the lung samples in the case of this study did kind of follow what we knew for soils. So for Tucson, 10% of the soil samples were positive. We did that work. But in Mesa, we know, because of other work in the lab, that 43% of the soil samples are positive. And so we do see more lung positives in Mesa than we did see in Tucson, and that does make sense. Transient fungi didn't affect our analysis, so here I'm assuming that fungi that never been reported to be associated with animals are probably transient fungi, and I get the same results when I take those genus out or if I keep them. Finally, our results do support the endozoan hypothesis that cocci is not causing coccidiomycosis, as there is no effect of Coccidioides in the microbiome, and that do align with another study in the literature. Limitations, we do acknowledge this study has limitations. This is a cross-sectional approach. We do have a small sample size, and I think the biggest one and the one that maybe is more tangible to try to work in the future is we don't know very much about those samples up until they were donated to us. So I think future studies, or even ourselves as we move on trying to get more and more data, try to work with like farms and zoos and wildlife monitoring programs to obtain samples that we know more of the general state of those animals. Thank you for your time. And yeah, it's amazing.
All right, thank you, Anna. So we're open for questions.
Thank you for a great job.
Thank
So,
you.
You're suggesting there was no cocci before mammals? Because I'm a little confused. Some of us think that this cocci was under the ocean. There was no mammals under the ocean.
Yeah, but they are still breathing air.
Under the
So,
ocean?
well, dolphins.
I mean,
Sorry, I don't know if I understood the question.
okay. So the dolphins were,
Yes.
the reservoirs were cocci?
Oh no. They had coccidioides.
No, I know they have, but evolutionary. Was there a cocci before mammals were actually, it evolved?
I do not know.
Okay. Thank you.
Sorry. Thank you.
So did I understand you correctly that in lung samples, you were finding evidence, tags for other fungi?
Correct.
So how does that address this idea that cocci goes resident in the lungs, and then when the animal dies, it's the first kid on the block to have at the dinner table, because all of these other fungi are at the dinner table too, and some of those grow faster than cocci.
Right. I don't think we answer that at all with this study, right? I can't really tell you that cocci would grow faster. That's what the hypothesis is. All that that we've seen is that there's no difference, and following the logic that if there is disease, there would be a change in microbiome. We don't see that. So, but I can't really test that. Ideally, we would be able to, I don't know, we talk about that in the lab sometimes, just having some mice on cages and, doing the ketamine and then just waiting to see if cocci grows faster, culture. But I'm not addressing that question at all. With this study, at least. Who knows in the future, right, boss?
Go ahead, and I just,
No, it's okay.
no, you go first. There's one on the line that I want to get to.
I'm popular, it sounds
You
like.
are. You are. That was a really fun talk. I appreciate it, and I appreciate the work you're doing in that light. In setting up some of your future experiments, or even maybe looking at the small data set you have, in humans, most people who get infected are never symptomatic, and you wouldn't even know they got infected unless something else happened. So there's nothing saying, without histology, there's nothing saying that these animals didn't have a controlled infection that was a granuloma.
Absolutely.
That now you're picking up the signature. So I'm a little, is there any correlation between the cocci burden in your sequencing data and something else about the animal, either other, in other words, you would think that the animal that had a fulminant infection and died from it would have a very high cocci burden, and that might change the microbiome.
But we don't have that data.
And Anna, because that's the online question,
As well.
Is this binary, or are there concentrations?
No, so those are just like DNA sequences.
But you showed the data. I mean, you showed graphs showing.
Right. So that's the abundance of DNA sequences or different genera in the sample. So all we have is the amount of DNA. So you can correlate that with the amount of organism, but I don't have fungal burden data for that.
No, I know you don't. Would you hypothesize? I mean, you don't show what your spread is there.
Yes.
So you would think that an animal that died from something else might have a controlled granuloma versus an animal that died of cocci would have high burden.
Right.
And then how does that impacting the rest of the microbiome?
Yeah, I don't know. And because those are like in the field, Ahilomyces, for example, is a pathogen as well that is known. So that might be what's going on. It's just for those samples, I think ideally, we would also be able to do something like this in the lab and have cocci be the only, and then look at the microbiome, but in a controlled environment. And then we can do all of that. But as I said, there's other pathogens going on. So I don't really know if the difference there would be cocci or not. Those are all tricky, difficult questions, I really appreciate, and I'm sorry that,
Okay, one more question.
Hey, Anna.
Hi.
Just wondering about spatial heterogeneity in the lungs. Like, do you know where in the lungs you're getting the tissue to extract from? And is it possible you're either getting a granuloma or missing a granuloma?
No, this is whole lung.
Whole lung.
Uh-huh.
Cool. So
Yeah.
you're like homogenizing and then grabbing?
Mm-hmm.
Cool. Okay.
So if it's there, we picked up on it.
Yeah, that's awesome. Thanks.
So Neil, one more point to follow up on his question. So it's the same question. Is it conceivable that you could do histopathology on those lungs and still have sample to study, like dividing it in some way?
Yeah, so we could, in the future, we have more samples that we're gonna try to put through the same pipeline. And we could use some of it for histology and some for DNA. So that's a good idea. Thanks, everybody. Sorry for the trouble here. I guess,
There's no trouble.
small mammals.
There's no trouble,
so
but
cool.
you're done. We have to move on. All right. Our next talk is pediatric coccidioidomycosis and an integrated management care system by Sean Fitzwater of Kaiser Permanente, Southern California, and also from the Valley Fever Center for Excellence at the University of Arizona.